Hello students, good morning. Children, today we are going to talk about biomolecules, and under the heading of biomolecule, actually we are going to discuss about polysaccharides, nucleic acids and nature of form. So these three topics today we will discuss. So we will start our discussion with the first important topic, that is polysaccharides. So polysaccharides are also known as glycans. These are a class of organic compounds which are long polymer of monosaccharides, right? That thing we have already discussed. And all these monomeric units join together by glycosidic linkage. Children, polysaccharide consists of more than 6 molecules of monosaccharides. They later are joined together by glycosidic bonds with loss of water each time, and a monosaccharide molecule is added. हर बार H2O का लॉस होता है जब मोनोसैकेराइड ऐड होते हैं, right? And simply we can write this equation as n number of C6H12O6. They give C6H10O5 N plus H2. So whenever there is a linkage of glycosidic bond, it is with a loss of water each time whenever there is addition of monosaccharide molecule, right? Now children, these are the most commonly encountered carbohydrate in nature. They mainly act as the food storage or the structural material. Talking about some of the example of polysaccharides, so examples are starch, cellulose and glycogen. Now let us move ahead with the types of polysaccharide. Children, the polysaccharides are of two types. The first one is Homopolysaccarides and second one is Heteropolysaccharides. Talking about the example of homopolysaccharide, that is the cellulose, an example for heteropolysaccharide is chitin. Now children, let us look about the first one, that is homopolysaccharide. So children, talking about the homopolysaccharides, these are composed of normal monosaccharide molecules of a single type. In homopolysaccharide, homo means same, polysaccharide is a polymer. So homopolysaccharide is a polymer of same kind of monomeric units. And children, basically they include three important biological substances. These are glycogen, starch and cellulose. All these are made up of by glucose unit, but differ in their arrangement of their bonds. So first of all, let us discuss about glycogen. So talking about the glycogen, it is the main reserve food material of animal cell. That is why it is known as animal starch. Because children, in plants the reserve food material is starch. So glycogen is the reserve food in animals, so that is why it is known as animal starch. Its molecule is a long, much branched chain of about 30,000 glucose units. So it is very high number of glucose units which are linked together. It is clearly visible in this picture also. And children, each unit is joined together by 1,4-alpha glycosidic bond, right? And it usually formed and stored in the muscle cell and the liver cell. Children, there is a process of formation of glycogen from the glucose. That process is known as glycogenesis. Glycogenesis is the process of glycogen synthesis in which glucose molecule are added to a chain of glycogen for the storage. So another important process which we will discuss is the glycogenolysis. So glycogenolysis is actually the breakdown of glycogen into the glucose, or we can say in the process of glycogenolysis there is the sequential removal of glucose monomer, and the process is known as phosphorylation by the enzyme glycogen phosphorylase, right? Children, talking about the liver of adult human. So liver of adult human stores 0.91 kg of glycogen, right? So that is why it is known as reserve food material. Now the next one is the starch. Now let us discuss about the starch. Children, it is a reserve food material of plant cell. It is also known as amylum. Children, the word amylum comes from the Greek language, and the Greek word is amylon, which means starch. So it is found in abundance of seeds and legumes. So here on your slide, here on your screen, there is a series of starchy food. It includes bread, cereals, pasta, rice, potato, beans, etc. Right? And starch consists of two components. There are two important main components of a starch. First one is amylose, which around forms 20 to 30% of a starch. And second one is the amylopectin, and rest all the starch is known as amylopectin. And children, both are glucose polymer. Let us take the example. Children, when potatoes are boiled, water becomes cloudy because of amylose dissolved in it, and amylopectin that remains in the potato. आपने देखा है जब पोटैटो को आप बॉईल करते हो पानी में तो वाटर जो है वो क्लाउडी हो जाता है, तो स्टार्च का वो पार्ट जो वाटर में डिसॉल्व हो जाता है वो कहलाता है एमाइलोज, और बाकी का बचा हुआ पोटैटो, रेस्ट ऑफ द स्टार्च इज एमाइलोपेक्टिन, और वो जो उबला हुआ आलू होता है वो क्या कहलाता है, एमाइलोपेक्टिन स्टार्च कंटेन करता है. Now the third homopolysaccharide is cellulose. So let us quickly discuss about the cellulose. Children, it is the most abundant carbohydrate, in fact most abundant organic compound in nature, right? It is the main structural polysaccharide of the plant. Cellulose molecule is a long branched chain of about 6,000 glucose units. And children, several molecules of glucose polymer are held together by hydrogen bond to form cellulose microfibrils, which are arranged in larger bundles, that is known as macrofibrils. It is clearly shown in the picture. So cellulose is actually the homopolysaccharide. It is known to be a rigid and insoluble polysaccharide found in the cell wall of most of the plant cells. Talking about the paper made from pulp of the plant and cotton fiber are also contain cellulose. They are also made up of cellulose. As cellulose is not composed of complex Higgs, so it cannot hold iodine and cannot give any color with iodine, right? Whether they are the category of homopolysaccharide, but they cannot give the color with the iodine, right? And the structure of the cellulose is on your screen. Now the next category which we will discuss is the heteropolysaccharide. Children, heteropolysaccharide are composed of modified monosaccharides molecule. These are the complex carbohydrate formed by the polymerization of two or more than two type of monosaccharide monomers. Talking about the example, chitin is the very good example. So chitin is the second most abundant natural polymer found in the exoskeleton of arthropod. Children, have you ever touched any kind of arthropod? Like if you talk about the example of cockroach, it is very hard from the outside. Why? Because it consists of exoskeleton which is made up of chitin. And children, chitin is also present in the cell wall of fungi. You have studied in class 9 also, as you have studied in your earlier chapter also of class 11, then the cell wall of fungi is made up of by chitin. And children, it is the building block of amino sugars, and chemically it is a modified sugar. Now let us talk about some of the important functions of polysaccharides. Children, polysaccharides play multiple functions and can be used in the following ways. The very first important function of the polysaccharide, that it acts as the structural compound. It acts as the structural compound because cell wall of plant is made up of by cellulose. Fungal cell wall and protestant cell wall is also made up of by the cellulose and chitin. Right? So that is why it is one of the very important structural component. So it is the very important function for the polysaccharide. Children, it helps in the anticoagulation and prevent blood clotting inside the vessels. For example, heparin. Heparin is the polysaccharide which basically helps in the coagulation of blood and prevent the loss of blood. The another very important function that they help in the lubrication of the joint between the bones. Talking about the example, high hyaluronic acid. So high hyaluronic acid is present in between the bones. It is present in the joints which actually lubricate the joint. It also used in the tissue culture, agar medium. It is used as the medium in the tissue culture. It acts as the reserve food material. For example, starch and glycogen, they act as the reserve food material. So this is about the polysaccharide. Now moving towards the another important topic, that is known as nucleic acid. Now children, let us talk about the nucleic acid. So let us first discuss about the history of nucleic acid. Children, talking about the nucleic acid. So nucleic acid was first isolated in 1868. It is first isolated in 1868 by a Swiss physician named as Frederick Mesher. From the nuclei of pus cell, he actually obtained the DNA. He actually obtained the nucleic acid. Children, because they are found from the nucleus, that is why these substances, then he named those substances as nuclein. क्योंकि उन्हें वो न्यूक्लियस से मिला था, क्योंकि उन्होंने उसे पस सेल के न्यूक्लियाई से ऑब्टेन किया था, तो इसलिए उन्होंने उस सब्सटेंस को नाम दिया nuclein. Now he found that nuclein is the substance responsible for the transmission of heredity character, right? And in around 1889, Altman renamed nuclein as the nucleic acid. Why he renamed the nuclein? Because children, he discovered the existence of nucleic acid. He discovered the chemical nature of the nucleic acid actually. He found that nuclein are the substances which actually responsible for the heredity of character, but chemically they are acidic in nature. So he renamed nuclein as nucleic acid. Now let us first talk about the locations. So children, nucleic acids are strong acids and were originally found in the nucleus. Talking about the composition, right? So the nucleic acid are giant molecule having complex structure and very high molecular weight. Basically, nucleic acids are polymer of nucleotide. Nucleotide are the building block of nucleic acid. That thing we have already discussed, children. The adjacent nucleic, and if we talk about the composition of nucleotide, as we have already discussed, it is made up of by heterocyclic compound, that is nitrogenous bases. 5 category of nitrogen base are there: adenine, guanine, uracil, cytosine and thymine. Then it is also composed of monosaccharide, which is the pentose sugar. 2 category of pentose sugar are there: ribose and deoxyribose. And the third important component of nucleotide is the phosphoric acids, that is phosphate group. Children, nucleic acid which contain deoxyribose sugar is named as deoxyribonucleic acid, while which contain ribose sugar is termed as ribonucleic acid. And children, the adjacent nucleotide are held together by phosphodiester bond, right? Means पहले न्यूक्लियोटाइड, ये चीजें हम डिस्कस कर चुके हैं पिछली वीडियो में। पहले न्यूक्लियोटाइड के जो शुगर होता है उसके 3 प्राइम कार्बन पे और सेकेंड एडजेसेंट न्यूक्लियोटाइड के 5 प्राइम कार्बन पे एक बॉन्ड क्रिएट होता है जिसे फॉस्फोडाइएस्टर बॉन्ड कहते हैं, राइट? नाउ चिल्ड्रन, देयर आर 2 टाइप ऑफ न्यूक्लिक एसिड्स. First one is the DNA. DNA is named as deoxyribonucleic acid. DNA is the genetic material and forms molecular basis of heredity in all the organisms. In certain organisms like virus, tobacco mosaic virus for example, RNA is the genetic material. So the second category of nucleic acid is RNA, and the structure of DNA is on the top of your screen. Now talking about the RNA, so RNA named as ribonucleic acid, it helps in polypeptide synthesis, like it helps in the protein synthesis, and it also conveys the genetic information but cannot self-replicate itself. Children, talking about the structure of RNA, the picture is on the top of your screen, right? Now children, talking about the structure of nucleic acids, so DNA is the double-stranded structure as shown on your screen, and RNA is the single-stranded structure as also shown on your screen. So this is about the nucleic acid. Now let us talk about the nature of bonds in polymer. Now children, let us enrich our knowledge about nature of bonds in polymer. Children, bonds which linking monomers. So in a polymer, successive monomers are linked by the specific type of bonds. And this bond formation occur by the dehydration reaction. That is, the reaction involve elimination of water moiety. जहाँ bond formation होती है वहाँ पे there is the loss of water moiety. Children, we studied polymers, like we have studied about proteins, sugars, nucleic acid, right? So first of all, let us discuss about the different categories of bonds, right? So first of all, we will discuss about the proteins. What kind of bonds are formed in the protein? So these are the different type of bonds, like peptide bonds, which are in between, which basically form the proteins, glycosidic bonds, which form sugar, and phosphodiester bond, which form nucleic acid. First of all, we will talk about the polypeptides, that is protein. Children, in polypeptides, successive amino acids are linked together by peptide bond. So here is the example, here is the picture of peptide bond. And the bond formed between the carboxyl group of one amino acid and amino group of the next amino acid with the elimination of water. So here it is clearly visible that between the successive amino acid, पहले अमाइनो एसिड का कार्बोक्सिल ग्रुप और दूसरे अमाइनो एसिड का अमाइनो ग्रुप के बीच में वाटर का एक लॉस, एक वाटर मॉलिक्यूल का लॉस हो रहा है. देयर इज द एलिमिनेशन ऑफ वाटर मॉलिक्यूल एंड फाइनली द पेप्टाइड बॉन्ड इज फॉर्म्ड. सो द पेप्टाइड बॉन्ड इज रिप्रेजेंटेड, basically it is represented by CONH, right? So here you see the peptide bond is formed, right? The next category which we discuss is the glycosidic bond. So here on your screen is the picture of glycosidic bond. Children, in polysaccharides, monosaccharides are linked by 1,4 glycosidic bond. And children, this glycosidic bond formed between the first and the fourth carbon atom of the successive monomeric unit. मोनोमेरिक यूनिट के पहला कार्बन और फोर्थ कार्बन देख रहे हो आप यहाँ इस पिक्चर में. फर्स्ट कार्बन और फोर्थ कार्बन के बीच में जो बॉन्ड फॉर्मेशन हो रही है वो ग्लाइकोसिडिक बॉन्ड है. एंड इन अ ब्रांच्ड पॉलीसैकेराइड, ब्रांचिंग इज बाय 1,6 glycosidic bond, right? So that is structure, and again here is the loss of water molecule, dehydration process is there. The next category which we will discuss is about the nucleic acid. What kind of bond is formed between the nucleic acid? It is phosphodiester bond. Children, in the nucleic acid, the successive nucleotides are linked by a phosphodiester bond. That is linking of 3rd prime carbon of the sugar of one nucleotide and 5 prime carbon of the sugar of next nucleotide. Okay, that thing we have discussed. So the bond formed between phosphate and hydroxyl group of sugar is known as ester bond. शुगर और फॉस्फेट के बीच में होने वाला बॉन्ड क्या कहलाता है? एस्टर बॉन्ड. उसी तरीके से फॉस्फेट और दूसरे न्यूक्लियोटाइड के शुगर के बीच में बनने वाला बॉन्ड एस्टर बॉन्ड. एंड सच एस्टर बॉन्ड ऑन ईदर साइड ऑफ द फॉस्फेट मोइटी. ये फॉस्फेट मोइटी के दोनों तरफ होते हैं. तो ये जो फॉस्फेट मोइटी है ये 2 एस्टर से जुड़ रहा है, इसलिए इस बॉन्ड को कहते हैं फॉस्फोडाइएस्टर बॉन्ड. राइट? Now children, again one category of bond I want to discuss with you. It is the hydrogen bond. Now children, you have discussed about, we have already discussed about the nucleotide. In that we have discussed about the structure of DNA and we have discussed that there are 2 strands, right? So it contains of nitrogen bases. तो एक स्ट्रैंड के नाइट्रोजन बेस ये आप देख रहे हो ये, और दूसरे स्ट्रैंड का नाइट्रोजन बेस ये, इनके बीच में जो बॉन्डिंग हो रही है वो कहलाती है हाइड्रोजन बॉन्ड. ये आप देख रहे हो ये बॉन्डिंग हो रही है. That is known as hydrogen bond, right? So this is about the nature of bond. So children, today we have discussed about the polysaccharides, we have also discussed about the nucleic acid, and we have discussed about the bonds, different bonds which are formed in the formation of polymers. So I hope you got the today topics. So children, now your task is to revise and learn today's topic. So very soon we will meet again. Till then, have a nice day and thank you.